Hello friends, good morning. Today we discuss about proteins. Okay, the structure, function and composition of proteins. Okay, totally it's about proteins. How the proteins were useful in our daily life and how these proteins, these biomolecules will generally have their impedance in every living organism. Okay, let's discuss about the proteins. Protein are almost abundant organic molecule of the living system. Yes, we have to agree with that. They consist of about 50% of the cellular dry weight. Yes, they consist of fundamental basis of structure and function of life. In 1839, Dutch chemist G.J. Mulder was first to describe about the proteins. That's past. The term protein derived from a Greek word proteos. Okay, it's proteos. It's a Greek word actually. So, and the proteins are nitrogenous macromolecules that are composed of many amino acids. Yes, the proteins, the basic structure of protein is amino acid. Amino acids, polymers are nothing but proteos, means proteins. Okay, here we see a 3D structure. It's a 3D structure of a protein. Here there are alpha helixes and beta sheets. We can see clearly here. Okay, these thread-like structures were beta sheets. These thread-like structures, and these ring-like structures were alpha helixes. But the both combination called as tertiary structure of protein. So this is the example of how the protein. Okay, and here we can see now orange color I highlighted. These are beta sheets. This sheet like structure of beta sheets and helix like structure is called alpha helixes. Okay, what are amino acids? As we described that the proteins were made up of amino acids, right? Amino acids, they are a group of amino acids. When they link up with each other by hydrolysis, means there is an outcome of H2O, water molecule, COOH plus OH, then minus H2O, they form COOH. Okay, usually how the amino acids will aggregate together to form a proteins. Okay, amino acids plays a very important role. They are the basic. The amino acid was a basic component of the protein, means basic fundamental structure. Without amino acids, how can a protein build? No, it's not possible usually. So let us discuss the amino acid, how amino acids look. There will be an amino group means NH3. There will be a carboxyl group means COH. There will be an R group means anything, any organic molecule can attach R. R is a normal group, means in the place of R there will be CH or CH3OH, anything. Okay, and there will be H group. Okay, this is the alpha carbon. Okay, so usually normally this is actually it is COO minus. Here it is COO minus, the NH3 plus COO minus. It is a form of, like, actually it is called as a deuterion, means neutral. Okay, where the ions are represented, means the minus and plus were represented, so it is called as a deuterion. Usually the basic structure of amino acid is COOH, and here NH3, here H, here some R group, any group. When it is combined with another NH3, okay, here there is loss of minus H2O, thus forms a peptide bond. Usually that forms a peptide bond. Here it forms like COH, N, H, H, and here CHR. Okay, here this is the peptide bond. Usually it is called as a peptide bond. Clearly it is a peptide bond. Okay, and these are some angles. It's called as phi angle, psi angle. Will be when we discuss Ramachandran plot or any 3D structure of the proteins, we discuss about these angles. pi angle and psi angle. Based on that, the alpha helix, beta sheets were there, and also the 3D structure is possible. Okay. Now discuss about some points about the amino acids. Amino acids are the group of organic compounds containing two functional groups. One is amino group and another is carboxyl group. Here we can see clearly, okay, we can see clearly here it is an amine group and it is a carboxyl group. Okay, here also it is an amine group, it is a carboxyl group. And the amino acid group NH2 is the basic, while the carboxyl group is acidic in nature. So it is quietly neutral, because one group is acidic nature and one group is basic in nature. There are about 300 amino acids occur in nature. Only 20 of them occur in proteins. Only 20 of them occur in proteins. Means usually in our body some proteins were manufactured by our body only. Okay. Some proteins by cell machinery, by transcription, by untranslation process. Some proteins were manufactured by our body. So those were nothing but mostly enzymes which were useful for metabolic processes. Generally for the uses of metabolic processes in our body, some proteins were manufactured inbuilt in our body by our cells by transcription and translation. From the DNA of the cell to the mRNA, that process is called transcription, and from the mRNA to proteins, that process is called translation. So by those processes, some proteins were manufactured in our body only. But in the proteins which are manufactured in our body, the amount of amino acids, the number of amino acids were only 20, but there are about 300 amino acids occur in nature. That's the meaning of that point. Okay. Next we go for the structure of amino acid, the clear-cut structure of amino acid. As I discussed earlier only, each amino acid had 4 different groups attached to alpha carbon, and this carbon is known as alpha carbon. The rounded carbon is known as alpha carbon. Okay, that carbon is known as alpha carbon. Each amino acid has 4 different groups attached to alpha carbon, which C atom next to COOH. These 4 groups of amino acids are COOH group, amino group, hydrogen atom and side chain. This R is nothing but a side chain. It can be a CH3, CH3OH, anything based on type of amino acid. And this is a amine group NH2, or it is called as NH3 minus NH2. Okay, and this is COOH carboxyl group, and this is hydrogen. That's the basic structure of amino acid. I think it is a NH3 here. One H should be there. Okay, so this is the basic structure of amino acid. There are different types of amino acids. As we described, there are 20 types of amino acids which are basic and also which are helpful for manufacturing proteins in our body too. And also most of the foods we take, they also consist of amino acids, mostly these amino acids only. So we take, or also some of the amino acids which are manufactured inbuilt. Based on that, there are 2 types of amino acids. One is essential and non-essential. The non-essential amino acids were the amino acids which are manufactured by our body only, and the essential amino acids were manufactured by, they are not manufactured. Essential amino acids we have to take from outside food. Daily we take food now, so the food which we take consists of those essential amino acids. So we have to choose food wisely, and we have to take food completely to essential amino acids, where the food has mostly more essential amino acids. So it completes the diet. Okay. Now we discuss about the types of amino acids. The protein amino acids, these are. So non-polar, aliphatic are groups. They are non-polar. First one is non-polar. I round up. Non-polar, aliphatic are groups. So here the polarity doesn't exist when in case of see, these are R groups basically. R groups. These are all R groups. So based on the change in R group, the type of amino acid also changes. Here, in case of glycine, the R group is hydrogen only. This is the basic amino acid. The glycine was the basic amino acid. So here the R group is hydrogen. And in case of alanine, the R group is methyl group CH3. And in case of valine, it is propyl group. And in case of leucine, it is 4 carbons, butyl group. And in case of methionine, there is S present in between this, and also methionine consists of sulfur. And in case of isoleucine, and this is the R group. So and next category we go for polar uncharged R groups. They are polar, but they are uncharged R groups. In case of serine, this methyl hydroxyl means this is the CH2OH. In case of threonine, this is the R group. In case of cysteine also, this consists of sulfur. This is the R group. These are polar groups, but they are uncharged R groups. And in case of proline, that's the R group. In case of asparagine, this is the R group. And in case of glutamine also, the R group is so complex. And positively charged R groups, those which are positively charged, here we clearly see that here the plus sign indicates those are positively charged. So positively charged R groups are lysine, and this is the whole is the R group. Even this, in this case also, this whole is the R group. Arginine, histidine, lysine were the positively charged R groups. And we go for negatively charged R group, aspartate, glutamate were the negatively charged R groups. Okay, negatively charged R groups, these two. This is the R group. R group was some other functional thing, means because based on R group only, the type of protein, the type of amino acid changes. And when we go for phenylalanine, they are non-polar aromatic R groups. Non-polar aromatic. They have aromatic rings. So non-polar aromatic R groups are phenylalanine, tyrosine, tryptophan. Phenylalanine, tyrosine and tryptophan were the non-polar aromatic R groups. Okay, let's see the groups. Okay, again, the non-polar were hydrophobic, generally alanine, valine, leucine and proline, phenylalanine, tryptophan, methionine. Those, as we discussed, and polar also we discussed. Acidic were negatively charged, aspartame, glutamic. These are some based on grouping. This is some basic classification. The positively charged, and these are all of total amino acids are 20. There are 20 amino acids. Okay, there are 20 amino acids totally, as we see in the screen. Okay, based on R groups, generally it varies. Everything varies. The characteristic of amino acid varies, and the basic function of amino acid varies, and different R groups were attached to different things to the basic structure of amino acid. So everything varies, and grouping of amino acid will form a protein. Okay, now we discuss about peptide bond formation. As I discussed earlier also, the peptide bond formation was a very important thing to form a protein. Means to establish a structural form of protein, how the peptide bond formation is very important, because one protein, suppose here we can see, if this is amino acid, okay, this is the COOH group and this is the NH3 group, and this is H, here R. When combined with another amino acid like NH3, here C, H, R, COOH, here this H and this H and this O, they combine to form minus H2O. The water is released, and this type of bonding is called peptide bond. Okay, this is called peptide bond. Usually it results to form like this, CNH3 H, here CON, because here H is eliminated and HO is eliminated, N H C H R CO minus. So here this bond is called peptide bond. Okay, that bond is called as peptide bond. Okay, alpha carbon, here we can easily clarify in this diagram. We can see here this is the CO. Okay, I am rounding up the CO in orange color. This is the CO, this is the CO. Okay, CO, this is alpha carbon, one is alpha carbon and one is here alpha carbon is a carbon where the function groups are arranged as called as alpha carbon. So this is the peptide bond. Alpha carboxyl group of one amino acid with side chain R1 forms a covalent peptide bond with alpha amino group of another amino acid with the side chain R2 by removal of molecule of water. It's very important. By removal of molecule of water, this results in dipeptide. This is called one peptide bond, or two amino acids were joined to form a peptide. The dipeptide can form a second peptide with a third amino acid to give a tripeptide. It's a chain: tripeptide, tetrapeptide, pentapeptide, hexapeptide. And repetition of this process generates a polypeptide or protein of specific amino acid has 40% double bond character caused by resonance. Okay, resonance also plays a very important role. Okay, which is the reason for 40% double bond character. Next, we go for detailed formation of peptide bond. As we discussed earlier also, before 2-3 slides also. Now we discuss about the detailed formation of peptide bond. Okay, here is amino acid 1, you see. Here is the amino acid 2. The both combine, and by removal of one molecule of H2O, water molecule, here the peptide bond was formed. This is the peptide bond. Okay, the blue color mark, the bond is called peptide bond. Okay, the peptide bond was so rigid it can't turn. The psi angle and phi angle means those where NH3C alpha carbon, those can rotate to form alpha helixes and beta sheets in case of secondary structures also. Okay, now polypeptide backbone in the repeating sequence of NCC is a peptide bond. That's a peptide bond. The side chain or R group is not part of backbone. Note, the side chains, the R groups were not part of backbones. Okay, here we can see clearly how the backbones were, how the peptide bond, the blue color marked ones were peptide bonds. These are the peptide bonds. These are the backbones of protein structure. Okay, overview of protein structure. Configuration: geometric relationship between a given set of atoms that distinguishes between L amino acid and D amino acid, dextro and levo. The spatial arrangements of atoms, the thermodynamically most stable configuration exists and stabilized largely by weak interactions, van der Waals interactions, those type of interactions. Okay, the stability is the tendency to maintain native configuration. Native configuration stabilized by disulfide bonds, non-covalent forces. These are involved to keep the protein in a native configuration. Native configuration means tertiary or quaternary structure, called as native configurations. The structure of protein in detail, we are going to learn the primary structure, the secondary structure, and tertiary structure and quaternary structure of a protein. So why these are so many structures means the primary structure was a basic thing. The primary structure, usually the basic thing, means the amino acids are arranged in a sequence. The secondary structure, which consists of normally alpha helixes like structure like this, helixes, or beta sheets. These are secondary structures usually. These are usually scientists were discovered how those alpha helices and beta sheets were formed, sheets like structures. And then tertiary structure is the combination of alpha helices plus beta sheets plus some usually some forces also they are involved. In the tertiary structure is the complex structure, and quaternary structure involves some magnesium and so on, so ions in middle, like a chlorophyll. Chlorophyll was the tertiary structure of it's also a protein, it's also a chlorophyll, also one of the enzyme type thing. And in case of hemoglobin example, hemoglobin also we express in quaternary structure. Quaternary structure is a very complex thing rather than the primary, secondary and tertiary structures. Usually we can see in the screen that primary structure was generally a line of sequence, and in case of secondary structures these are beta sheets and these are alpha helices. And in case of tertiary structure, this is the tertiary structure, this three-dimensional structure. And in case of a quaternary structure, it's a complex protein, usually the globular proteins or complex proteins. So what forces determine the structure? Okay, the primary structure determined by covalent bonds between the chain of these amino acids. Okay, and the secondary, tertiary and quaternary structure determined by weak forces like van der Waals forces. Those are the weak forces which are involved in between the atoms. Okay, the primary structure, this is the primary structure. This is a simple, the primary structure was a simple thing, simple one means this is a group of sequence like this, this is sequence just. And the secondary structure which consists of alpha helix and beta sheets, and the tertiary structure was placer sheet and alpha helix. Sheets and helices, sheets, beta sheets and alpha helices both tend to form the tertiary structure, and the quaternary structure is nothing but a very complex one, and it's a interaction. The interactive thing is quaternary structure, the final structure is quaternary structure. This is the steps like assembly, folding, packing, interaction. Okay, assembly, folding, packing and interaction. Okay, the next let's discuss about the primary structure. This is the primary structure. The primary structure is usually the primary structure of the protein refers to a sequence of amino acids present in polypeptide chain. It's a simple chain of amino acids. It's just a sequence like ASP, leucine, like that simple chain of different combinations of amino acids. It's a simple chain, a chain-like structure. A chain-like structure was called as primary structure. Let's read this. The primary structure of protein refers to a sequence of amino acids present in polypeptide chain. Amino acids are covalently linked by peptide bonds or covalent bonds. Each component of amino acid is polypeptide is called as residue or moiety. By convention, the primary structure of a protein starts from amino terminal. Amino terminal means N terminal and ends with carboxy terminal means C terminal. Usually like this, NC. This is primary structure, sequence of amino acids. And the secondary structure were nothing but those who discovered how these alpha helices and beta sheets were formed. Usually the secondary structure consists of alpha helices. Here the proteins are linked up like this. Here the proteins, example leucine, aspartame, the proteins are linked up like this. These pink dots were nothing but different chain of proteins. They are in form of helices. And in case of beta sheets also, the proteins were linked up like this in a sheet manner. Means these two lines means here one protein, one amino acid, one amino acid, one amino acid. These are amino acids, pink dots are. These are amino acids. These are amino acids. Okay, here the amino acids were linked by some weak forces. Okay, weak forces. Here also the amino acids were in a helix manner, linked by weak forces. So amino acids were linked with each other. Formation of beta sheets and alpha helices, which are further connected in the tertiary structure. Okay, It's locally occurring structure in the protein and also mainly formed through hydrogen bonds between backbone atoms. Here the backbone atoms, there's a hydrogen bonds in between hydrogen bonds. Here also in between these are nothing but hydrogen bonds. Hydrogen bonds are weak bonds, usually weak forces. The structures were formed. Pauling and Cole studied the secondary structure and proposed two conformations. One is alpha helixes and beta sheets. These are very important and structural components in case of a tertiary structure, because tertiary structure is full of this alpha helixes and beta sheets. Combination is nothing but a tertiary structure. And what is an alpha helix? Here we can see clearly this is one amino acid, this is one amino acid, this is one amino acid, like this is one amino acid. These were bonded by weak forces, or nothing but hydrogen bonds. Okay, right-handed spiral structure, alpha helix. Let's discuss about alpha helix. Side chain extended outwards, stabilizing by hydrogen bonds. This is very important. Hydrogen bonds were act as stabilizing agents that are arranged such that peptide carbonyl oxygen nth residue and amino hydrogen in n plus fourth residue. Amino acid per ton is 3.6 amino acids per 1 ton. 1 ton. This is the 1 ton. And pitch is 5.4 angstroms. Pitch, the gap is 5.4 between 1 ton. Alpha helix segments that found in many globular proteins like myoglobin and trophinin. Length equal to 12 residues and 3 helical turns. Phi angle is minus 60 degrees and psi angle is minus 45 degrees. Falls within the fully allowed regions of Ramachandran diagram. The Ramachandran plot explains about these structures, the alpha helixes and beta sheets, in a greater way. Usually phi angles and psi angles are those angles between the rotation of a CO group and NH3 group between a carbon atom is there. So this is the structure of usually in alpha helix. There is a number of residues, 3.6 residues per ton. And let's go for types of helices. There are 3 types of helices mainly. The alpha helix is also called as alpha helix, also called as 3.6. Okay, so hence we discussed before only the alpha helix, right-handed one. And in phi helix, the other type is very loosely coiled H bonding pattern n plus 5, rarely found in nature. Okay, rarely found in nature. Next one is 3 below 10 helix, 310 helix, very tightly coiled H bonding pattern is n plus 3, which is rarely found in nature also. Okay, what we observe here, in case of alpha helix, H bonding pattern is n plus 4. In case of phi helix, it is n plus 5. In case of 310 helix, it is n plus 3. So it's called as n plus 3. So these were the types of helixes. Next we go for a beta plated sheet. Means it's called as, it's not commonly called as beta sheet. Beta sheet structure, how it is formed. Let's discuss a beta sheet. Here this is one amino acid, this is one amino acid. Here in between there is a hydrogen bond, so it appears like a sheet. Okay, in case of alpha helix, the hydrogen atoms were linked between like a chain structure, vertical structure. Here also, here it is a horizontal structure. See, it is a plated. This is the chain like this. Okay, here at first there are 1, 2, 3, 4. Okay, this is the 5, 6, 7, 8 amino acids. The one amino acid is linked with 8th one, and second one is linked with 7th one, 4th one is linked with 5th one. Okay, by these hydrogen bonds, these are the hydrogen bonds in between. These are nothing but the hydrogen bonds. We act as the formation of a beta sheet. In case of beta sheet also formed with 2 or more polypeptide lines up side by side. Individual polypeptide beta strand, each strand is fully extended. They are stabilized hydrogen bonds between N, H and carbonyl groups of the adjacent chain, the next adjacent chain. Okay, the beta sheet comes in 2 varieties. Here also 2 varieties. In case of alpha helixes, there is alpha helix, phi helix and 310 helix. There are 3 types of helixes. And in case of beta sheets also, anti-parallel beta sheet, parallel beta sheet. And there's two types. One is parallel and anti-parallel. Number one, we discuss about anti-parallel. The anti-parallel, this is the anti-parallel beta sheet. And this is the parallel beta sheet. Okay, here we can observe clearly anti-parallel means one chain is going like this direction, the other chain is coming like another direction. This is parallel. When this is anti-parallel, first one is anti-parallel and the second one is parallel chain. One chain is going like this forward. To start another chain, start an adjacent chain, here it should start from here only, from this side only. So it is parallel. One is parallel, the other one is anti-parallel. Anti-parallel is continuous type. It's continuous. Here the loop is continuous. In case of parallel, the chain starts from the first onwards, from this side onwards. That's a major difference. Okay, let's discuss the lines. Anti-parallel beta sheets, neighboring hydrogen bonded polypeptide chain run in opposite direction like this and like this. One is like this and one is like this, anti-parallel. When in case of a parallel beta sheet, hydrogen bonded chain extends in same direction. Here the same direction follows. Okay, the connection between two anti-parallel strands may be just a small loop, but the link between tandem parallel strands must be crossover connection that is out of the plane of beta sheet. Okay. Okay, there are two major sorts of connections between beta strand. One is a hairpin or the same end connection, a crossover or opposite end connection. Hairpin or same end connection is nothing but anti-parallel beta sheet. Crossover or opposite connection is nothing but parallel beta sheet. Okay, the hairpin connection, here we can see the hairpin connection is continuous. Hairpin connection, this is the hairpin connection. When you come to right hand crossover connection, it cross over to opposite end connection usually. And here there is the two types of crossover connection. One is a right hand connection and the one is left hand connection. Right hand opposite connection and left hand opposite connection when it comes to crossover or opposite connections. Hello guys, let's discuss about dihedral angles. Dihedral angles, most of the people confuse about the dihedral angles. Dihedral angles is nothing but torsion angles, very important. It's the angles about the phi and psi angles between C alpha carbon. Many people confuse about this. These are very important to determine the secondary structures, means the alpha helixes and beta sheets, how they are organized is determined by these dihedral angles. They are very important. Usually, and also tertiary structure is related to these alpha sheets and alpha helixes and beta sheets. So dihedral angles, even for a Ramachandran plot, the dihedral angles, the phi and psi angles were very important. Okay, let's discuss about the dihedral angles. Dihedral angle is nothing but it's an angle between two intersecting planes. Okay, it's an angle between two intersecting planes. It helps to maintain the protein structure. Let's discuss in detail about the dihedral angles. Okay, dihedral angle here, in case of let me explain dihedral angle very clearly. Here, the angle between C alpha carbon and the angle between the C alpha carbon, this is alpha carbon, this is also C alpha carbon, C alpha carbon and N group is nothing but phi angle. Okay, phi angle, and the angle between C alpha carbon and CO group is nothing but psi angle. Let me discuss very clearly here. I drawn the structure. So this is one C alpha carbon and this is one another C alpha carbon. This is one amino acid. This is one amino acid. There are two amino acids. In between CO and H means it's nothing but peptide bond is formed. The both are linked with peptide bond. Both are linked with peptide bonds. The peptide bond is not rotatable. It is very rigid. We know that it's very rigid. The peptide bond is very rigid. And so where the rotation is done? The rotation is done basically in between the C alpha carbon. Here, between the C alpha carbon and N, the angle is known as phi angle, pi angle, the rotation. And between the C alpha and CO group, the angle rotation is psi angle. And here also, between the C alpha carbon and N group, the rotation is pi angle. And the rotation between the C alpha and CO group is nothing but psi angle. As we discussed here, the C alpha carbon and N group is nothing but pi angle. The C alpha carbon and CO group is nothing but psi angle. Okay, clearly I mentioned here pi and psi angle. First, in the left side it is pi angle. C alpha carbon, we have to remember what is C alpha carbon. The C alpha carbon where the R group present, R group. So where the function group or where the other group is added, means some secondary group like methyl group, hydroxyl group, to determine the nature of amino acid. So amino acid basically there are different types of amino acid because the R group changes. So the R group attached carbon or the R group and hydrogen attached carbon is known as C alpha carbon. In the left side of C alpha carbon means the C alpha carbon and N, the bond is the bond is rotated with an angle of those angle is known as phi angle, and the C alpha carbon and CO, that bond rotation is known as psi angle. Okay, I think this is clarified. This is very, very important. Most of the people confuse with this only. The phi and psi angles are very important. Let's go detail about the phi and psi angle. Okay, the backbone or main chain of a protein refers to atoms that participate in a peptide bonds ignoring the side chains of amino acid residues. Okay, the only reasonable free moment or rotation around the C alpha N is nothing but pi angle and measured as phi angle, and C alpha C is measured as psi angle. This is pi angle, that is psi angle, and measured as psi. Okay, these angles are both defined as 180 degrees when the polypeptide chain is a full conformation. The conformation of the backbone can therefore describe as a torsion angles, is called as dihedral angles or rotational angles. So how they rotate, so those were called as torsion or dihedral angle. So based on that, beta sheets, alpha helixes, or the structural those help in determining the structure, whether the protein structure is valid or not. How we know? By pi and psi angles, by these dihedral angles. The dihedral angles determines whether the protein structure is stable or it's not stable. Okay, now we discuss about Ramachandran plot. Ramachandran plot I will discuss in further lectures. So Ramachandran plot is very, very important. Further videos I will discuss again. Ramachandran plot to visualize the backbone of amino acids. So how the backbone, say how the protein, whether the protein or it is stable or not, whether the secondary structure is based on secondary structures, mainly alpha helixes. Ramachandran plot was mainly based on alpha helixes and beta sheets. How the phi, based on the phi and psi angles, this Ramachandran plot was there. So based on that, the stability can be determined. Ramachandran plot to visualize the backbone of amino acid residues. The amino acids with the larger side chains will show less number of allowed regions within the Ramachandran plot. The amino acids with larger side chains will show less number of allowed regions within Ramachandran. This point is very important. So this is the Ramachandran plot actually. Here in case of plus psi values, the beta sheets, and the right-handed helix, left-handed helix is shown. These are the phi and psi values, 0 to 180 degrees, 0 to 180 degrees. Based on the angles, we can determine where the beta sheet is located. Where the angles are correct, only the protein will be in stable position. So that's the use of Ramachandran plot to determine whether the stability of the protein. The Ramachandran plot is used. Okay, this is some detail about the Ramachandran plot. These are anti-parallel sheets. This region is anti-parallel sheets. As we discussed the anti-parallel sheets, though there were no crossovers. Anti-parallel sheets like this continuous string, and the parallel sheets were like this. Okay, collagen triple helix, suppose this is an example. Right twisted beta sheet also here in this region it will be there, and right-handed alpha helix. This is right-handed alpha helix, and this is left-handed alpha helix. So usually 180 degrees configuration, the bond can be rotated between the C alpha and N is nothing but phi angle. The C alpha and C is nothing but psi angle. A Ramachandran plot is a way to visualize the backbone dihedral angles. As we discussed, dihedral angles phi and psi of amino acid residues in protein structure. So Ramachandran plot can use it to show which values or conformations of the phi and psi angles are possible of amino acid residues of the protein, and to show the empirical distribution of data points observed in single structure. That's cool. And the darkest areas correspond to core regions representing the most favorable combinations of phi and psi angles. Okay. Let's discuss about glycine and proline, two amino acids. The glycine and proline has no preferred Ramachandran plot. Okay, why? Glycine is formally non-polar. It's very small side, as we discussed before only what are polar, what are non-polar. It's very small side chain, may have no real contribution to hydrophobic interactions, means the hydrophobic interaction to bind up. And the proline has an aliphatic side chain and with a distinctive cyclic structure. The secondary amino acid group of proline residues is held in rigid conformation, reduce the structural flexibility of the bond. So here we can notice in these two things, the glycine and proline has no preferred Ramachandran plot because they can vary, because the forces can vary. And next we go for protein folding. What is protein folding? We studied the primary structure, secondary structure, tertiary structure. Okay, primary structure is nothing but this one is the primary structure. After the secondary structures are nothing but beta helixes and alpha sheets. Alpha sheets and beta helixes. So alpha helixes and beta sheets with the secondary structures. Beta sheets are sheet-like structures and alpha helix means helix-like structures. Those were combined to form a tertiary structure. So by folding, usually the protein has to come to original configuration. Original configuration is nothing but tertiary structure. Usually the protein will fold the hydrophobic interaction, hydrogen bonds. So these are all help in folding of proteins. Okay, the peptide bond allows for rotation around it, and therefore the protein can fold into orient and R groups in favorable position. Means usually due to the phi and psi angles, when phi and psi angles are nothing but those various the CN and CO, they can rotate to form alpha helixes and beta sheets, and those combination will form a tertiary structure of protein. These all results protein folding. Significantly, the protein will fold after the formation of protein. Every protein should fold to attain the functional structure. The functional structures are nothing but tertiary and quaternary structure. Quaternary structure for some proteins and tertiary structure for some proteins. They are fully functional proteins. The primary structure is not functional and secondary structure is also not functional. Weak non-covalent interaction will hold the proteins in functional shape. These are weak and will be made to hold the shape. Protein folding occurs in cytosol. Cytosol means cell area, in the cytosol only, in the cell area only. The cell, because cytosol is nothing but cell inside the cell only, not outside the cell. So there is a protein folding. How the protein folds? By protein folding only, the secondary structure is converted to tertiary structure. Two regular folding patterns have been identified formed between the bonds of peptide backbone. Okay, here these are alpha helixes, the formation from primary structure to secondary structure. These are beta sheets. Alpha helix protein turns like a spiral, as we know, and beta sheets protein fold back into a ribbon like globular protein. Turns and loops. Let's discuss about the turns and loops. In addition to alpha helix and beta sheets, a folded polypeptide contains two other types. Secondary structure called in the secondary structure, those are loops and turns. Loops and turns. These are very important. These called loops and turns. Loops and turns connect a alpha helix and beta strand. So in case of when the secondary structure generally formed to tertiary structure, these loops and turns connect with each other to form tertiary structure. Here the concept is very clarity. The primary structure, after the primary structure there are secondary structures, the alpha helix and beta sheets. Those were connected with each other to form tertiary structure by turns and loops. The most common type cause of a change in direction of the polypeptide chain, allowing it to fold back and itself create a more complex structure. Loops have hydrophilic residues and they found in surface of proteins. Loops have only 4 to 5 amino acid residues called turns. They were integral they have internal hydrogen bonds. Reverse turns are form of tight and where the polypeptide chain makes 180 degrees change in direction. Reverse turns are always also called beta turns because they are usually connect adjacent beta strands in beta sheets. In case of anti-parallel beta sheet, we observe reverse turns. Okay. No, not anti-parallel. In case of parallel beta sheets, we observe reverse turns. These are called reverse turns in case of parallel. These are loops and turns were there to form a tertiary structure. That's a basic thing. These are beta sheets. Beta sheets are connected with loops. Okay. This is anti-parallel. This is anti-parallel beta sheet. Okay. This is anti-parallel beta sheet. So anti-parallel connected with loop. But in case of parallel beta sheet, they are connected with turns. Beta turns, also known as beta bends or tight turns. In beta turn, here the turn, because this is one sheet and this is one sheet, these are connected with a turn. It's called beta turn. How the beta turns are formed, clearly explained here. The hydrogen bond is formed here. You can notice, and glycine residue also there. So in beta turn, a tight loop is formed when the carbonyl oxygen of one residue forms a hydrogen bond with amino protein of amino acid, the 3 residue down the chain. This hydrogen bond stabilizes the beta bend structure. In beta turn, a tight loop is formed when the carbonyl oxygen of one residue forms a hydrogen bond with amide proton of the amino acid 3 residues down the chain. Proline and glycine are frequently found in beta turns. Okay, they are the two which proline and glycine, as we discussed, because they didn't have a proper Ramachandran plot. Frequently found in beta turns. Proline because it's a cyclic structure, ideally suited for beta turn, and glycine because with the smallest side chain of all amino acids is mostly sterically flexible. A beta turn means by which the protein can reverse the direction of the peptide chain. Beta turns often promote the formation of anti-parallel beta sheets. As I discussed before also, the anti-parallel beta sheets were formed by beta turns. Tertiary structure of protein, which is very important. There is a functional structure of protein. The tertiary structure of protein was a composition of alpha helixes and plus beta sheets. But both the integration with loops and turns results the tertiary structure of protein. Tertiary structure, it's a completely folded thing. The tertiary structure defines a spiral overall 3D shape of the protein. The tertiary structure is based on various types of interaction between side chains of peptide chains. We have to note there is a clearly when we see the tertiary structure of protein. This is the tertiary structure. We can see hydrophobic interaction for hydrogen bonds and salt bridges, helixes, alpha helix and beta sheets were alpha helix and beta plated sheets where interact, and also there are the disulfide bonds. These all complexly is called a tertiary structure of protein. The tertiary structure stabilization in globular proteins, the tertiary interactions were frequently stabilized by sequentiation of hydrophobic amino acid residues in protein core, and in secreted proteins disulfide bonds between cysteine residue help to maintain the tertiary structure. Means how they stabilize. Interactions stabilizing the tertiary structure. Okay, what were the interactions stabilizing the tertiary structure? Let's discuss this briefly. So disulfide bonds, hydrophobic interactions, hydrogen bonds, ionic interaction, van der Waals forces. These are the interactions where how the tertiary structure is stabilized. We can see here clearly these all hydrogen bonds, disulfide bonds and van der Waals forces, and there are so many here. So by these all forces the protein is folded from secondary structure to tertiary structure. Okay, tertiary structure disulfide bond forms a bridge, disulfide bridge. Okay, it's a covalent bond between sulfur atoms on two cysteine amino acids. Usually cysteine amino acids, the cysteine amino acids consist of sulfur. So these amino acids generally consist of sulfur. So both when the parallel with each other, they form disulfide bonds. Okay. Hydrogen bonds are generally formed because they are weak bonds. They are the reason for formation of hydrogen bond. In case of alpha helices and beta sheets, which are integration of a tertiary structure, by them only the alpha helices and beta sheets consist of hydrogen bonds. They are weak, okay, which can be broken and reformed easily, allow structural changes and produce the functional molecules. Hydrogen bonds are weaker bonds. And next, salt bridges. As we seen in the tertiary structure, salt bridges are also electrostatic bonds between oppositely charged groups. The strength is usually 4 to 7 kilocalorie. These are the salt bridges. You can see ions of R group form salt bridges to ionic bonds. NH3 plus and CO areas of the protein attract form ionic bonds. Okay, and then we see about tertiary structure hydrophobic forces, which are also useful for aggregation of a structure. The close attraction of non-polar R groups through dispersion forces. They are non-attractive interactions but results in inability of water to form hydrogen bonds with certain side chains. Okay, hydrophobic region, this is called a hydrophobic region. They are also very, very useful. Next we go for total overall discussion. What are the forces stabilizing interaction of tertiary structure? There are hydrophobic interaction, hydrophilic interactions, salt bridges, hydrogen bonds, disulfide bonds. These all were the responsible for stabilized tertiary structure. Stabilized structure, which are generally a functional protein, consists of stabilized structure. These all interactions are very, very responsible for protein aggregation or protein folding. Okay, for protein folding to tertiary structure. The below all forces were responsible: the hydrophobic interactions and hydrophilic interactions, salt bridges, hydrogen bonds and disulfide bonds. Okay, these are all responsible. Okay, let's discuss about globular proteins. Globular proteins fold up into compact spherical shape. They functionally include biosynthesis, transport and metabolism. For example, myoglobin is a globular protein. That's a quaternary protein. Quaternary is a packet of oxygen here. It carries oxygen. This is a myoglobin protein. Myoglobin. It carries oxygen. Generally, it binds to heme group. Oxygen binds to heme group. And fibrous proteins, when we generally consider muscles, those consist of fibrous proteins usually. Fibrous proteins consist of long fibers and maintain structural proteins, means alpha keratins. In case of hairs, they are keratins. Hairs, fur, nails, skin, they are very strong because of these fibrous proteins. And beta keratins are the fibrous feathers and scales. Those are fibrous proteins. Super secondary structure. What is super secondary structure? Association of secondary structures. Okay, the beta sheets super secondary structure, alpha helix super secondary, mixer super secondary. So these associations were the result of tertiary structures. Okay, let's B and B unit, 4 alpha helix, beta meander, Greek key, polar helices, zinc finger, leucine zipper and beta barrel. These are some super secondary structures. In case of secondary structure, generally the secondary structures are nothing but alpha helices and beta sheets. So these are super secondary structures. Okay. In case of there are many types of super secondary structures. Next, we discuss about domains. A domain is a basic structure unit of protein, distinct from those make up the conformations. Part of protein that can fold in stable structure independently. Different domains can impact different functions of protein. Okay, this is a new concept, the domain. A domain is a basic structure unit of protein that distinct from those make up of conformations. Okay, these are very distinct. Next, we discuss about myoglobin. As already we discussed about myoglobin. Myoglobin is a small monomeric protein which serves as an intracellular oxygen storage site. They store oxygen. This is the beta turn. We can clearly see the structure of this myoglobin. Beta turns, alpha helixes, heme group. Okay, this is NH3 turn, NH3 side, this is COO side, this is alpha helix. The alpha helix and beta structure, this is a tertiary structure. No, we can consider it as a quaternary structure because heme group is present. It's a quaternary structure of hemoglobin. It's a not a hemoglobin, it's a myoglobin. Quaternary structure of myoglobin. So quaternary structure of protein. The quaternary protein structure involves the clustering of several individual peptide or protein chains, final specific shape, a variety of bonding interactions including hydrogen bonds, salt bridges and disulfide bonds hold the various chains of particular geometry. Okay, usually the tertiary structure also consists of hydrogen bonds, even hydrophilic and hydrophobic hydro bonds, and also disulfide bonds, salt bridges. Those all. There are two kinds of okay, let's discuss about the quaternary structure. There are two kinds of quaternary structures. Both are multi-subunit proteins. Homodimer, association between identical polypeptide chains, and heterodimer is the interaction between subunits of very different structures. Two types, one is homodimer and heterodimer. The interactions with the multi-subunit are the same as found of tertiary and secondary structures. And this is the complex structure of quaternary protein. The quaternary structure adds stability of decreasing the surface volume of the ratio. Okay, it is very folded, highly folded thing is quaternary structure. It is also a functional protein. Simplifies the construction of large complexes, viral capsids and proteasomes. This is the quaternary structure. And subunit and symmetrically arranged. This is symmetry about the symmetry, cyclic symmetry, and the other type of dihedral symmetry, how the symmetry, the foldation and rotations of the proteins. This is about the hemoglobin. Previously we discussed about myoglobin. Hemoglobin is a globular protein with 4 polypeptide chains bonded together. The 4 heme groups which bind the red blood cells for oxygen. Okay, hemoglobin is hemoglobin which is present, it's a protein which is present upper surface of red blood cells. Red blood cells are concave structure, so to imbibe the hemoglobin group, to imbibe the oxygen much more, the hemoglobin, the heme group, one heme group binds to 4 oxygen molecules. Okay, there are 4 heme groups and each contain iron. Each heme group can carry 1 oxygen molecule, so 4 heme groups can carry 4 oxygen. Here the concept is very clear. Each hemoglobin consists of 4 heme groups. Each heme group can carry 1 oxygen. So totally 4 heme groups can carry 4 oxygen molecules. Okay, we can see here the iron is present. Heme group, and this is a beta chain, this is alpha chain. There's a totally structure, how the structure is there. And next, these are the key concepts. The tertiary and quaternary structure result of folding of primary structure. Yes, by the folding of protein results the tertiary and quaternary structure, the 3-dimensional structure. The tertiary structure, mainly most proteins tertiary structures are a combination of alpha helixes and beta sheets and loops and turns. It's a very important point. The tertiary structure consists of alpha helixes, beta sheets and loops and turns. Larger proteins often have multiple folding domains. Okay, domains are irrespective of the configurations. And folding of H2O soluble globular proteins into the native structures follows same basic principles, minimizing the solvent accessible surface area and those all, maximizing the intra-protein hydrogen bonds, chilarity, very important. And the quaternary structure, some proteins have multiple polypeptide chains. Quaternary arrangement of polypeptides is multimeric proteins in general symmetry. Quaternary structure can play important functional roles of multiple subunits and especially in regulation also. Okay, now this is the overview of how these are the alpha helixes, beta sheets. Those form the tertiary structure and those form the quaternary structure. The more combination of tertiary structures, one or more tertiary structure can form quaternary structure. This is the how the protein usually folds. This is all about the protein folding. In case of protein folding also, we can write this from primary structure to secondary structure to tertiary structure to quaternary structure. So this all process is called as protein folding. Usually protein folding, generally proteins fold. Why proteins fold? Because usually the configuration, the stability, higher stability will have in quaternary or tertiary structures only. So functional proteins are nothing but they are tertiary and quaternary structure. They can be functional. Irrespective of primary structure can be functional. So every protein folds. Okay. The summary of structural levels: the primary, secondary, tertiary and quaternary. The sequence of amino acids: the secondary is the coil of alpha helix and beta sheets. Okay, they are secondary. And tertiary is the folding of protein into compact into three-dimensional. It's a tertiary. Quaternary, the combination of two or more protein subunits from larger biological active protein. This is quaternary. Thank you so much for listening. Signing off, yours Madhu. So if you have any doubts, please comment, subscribe for more videos. Okay, signing off. Bye, take care, see you.